those queen cell cups have got to be full of royal jelly, and you'll see why. Down here, Dr. Joe Laksha gave me some photos some time ago. These are mature queen cells, and look at the royal jelly. Royal jelly is a keystone to good, large, healthy queens. Last year, we were at the right place at the right time. On the upper right, that mite on the bee in the abdomen, and it was chewing the body fat off that bee. And we have a video of it, and you can see the intestines of the mite as it was eating the body fat off that bee. So really what we're saying here, we have 40-hour queen cell technology genetics fighting the mites, and that's where we're at with all this. Down here in lower left, this queen cell, beautiful queen cell, that queen came from this bar of queen cells. Her name is Hope, was Hope. She was produced in 2013. I named her Hope. Only time I named the queen. Why? Because back in 2013, we didn't have a lot of hope. We had a lot of problems. And Hope is in the logo of the Heartland Honeybee Breeders Cooperative. She lived for 3 years before she was superseded. And her sister, Hope's sister, was also. That's why we want to produce good, healthy, large, well-mated queens. There's two sides of the queen. You have one side that she can lay unfertilized eggs. And the unfertilized eggs is only reflected from the queen and her parents. It's not fertilized. So it is a direct descendant of the queen's mother and father through the queen. And that's a big deal for what we're trying to do. And if you have a strong, healthy colony, and with a little help with some brood foundation, you can produce anywhere from 3,000 to 5,000 drones during the summertime. You want those drones, those genetics out there flying around trying to find a DCA to mate with a queen. On the right-hand side, you have the fertilized eggs that the queen is fertilizing the eggs as they come down through her ovules. And she lets about 5 sperm out from her spermatheca to fertilize that egg. We would like to have her mate with at least 15, a minimum of 15, up to 30 drones. And a lot of research lately, they're finding that there's more and more drones are mating with good, healthy queens in good gene pools. So that's the two sides of the queen. One side, she's fertilizing eggs for workers. And some of those fertilized eggs could become queens. On the other side, you have unfertilized eggs, and those drones are the ones we want to fly out there and get into the drone congregation area to mate with good feral queens. About Dwight, I'm president of West Central Ohio Beekeepers Association. I'm director of OSBA, top of Ohio region. I have 10 counties. I helped found the Heartland Honeybee Breeders Cooperative in 2013. I work with Purdue, Penn State, and OSU. And Zell and I just started the Ohio Queen Bee Improvement Enterprise down at Zell's place with feral swarm trappers and Queen Production Network. And that's where you guys come into. I'm on the education committee, OSBA Education Committee. I do the monthly USDA honey report, and Roger is one of the guys that feeds me his information from where he lives up north of me. And then I have bees at Wright-Patterson, at Honda and Purdue, and you'll see that in a minute. And long term, what we're going to do with Honda, their goal is in a couple of years to provide nukes for the FFA and the 4-H scholarships. And you'll see a little bit more about that later on tonight. I'm also a member of the Ohio Pollinator Habitat Initiative. I do their Facebook page. And we have over 7,500 followers. It's made up of ODNR and ODOT and Fish and Water and Pheasants Forever, Quail Forever, ODA, Soil and Water. Basically, it was made for built for monarch butterflies, but honeybees and native bees, bumblebees, all the pollinators fall into that area. So that's my life. That's what I do all the time. I'm retired. I'm 80 years old. My wife passed away in 2009 from Alzheimer's, so I decided I wasn't going to curl up and wait to die. I wanted to do something, so here I am at 80 years old, and with a lot of irons in the fire, and I love it. Okay, this is something very interesting. 1976, Dr. Friedrich discovered Varroa in Germany and Austria. It had come up from Asia, and in 1987, he discovered that the mice were being chewed by bees over in that area. You'll see here in a little bit. Thank goodness he took some pictures of it. In 87 is when Varroa invaded North America. In 1992, Rutner published a paper, and thank goodness he had a microscope, and he has chewed mite photos in his paper. And they're exactly like what we're having chewed over here in the US. Dr. Greg Hunt, at the time, he was the principal geneticist out at Purdue University at the bee lab, found on chewed legs on a colony of bees south of Cleveland. His brother had bees and he got tired of them. He left the colonies behind his mother's house. One of them survived. Greg discovered they were chewing and took them over to Purdue. 2011, my buddy Price Smith at Champaign County saw mice being chewed by bees. So I gave him my HD video camera. This is before smartphones. And he spent several hundred hours doing about 15 different videos of bees chewing mites. 2012, I crossed paths with Tom Seeley down in West Virginia, and he talked me into starting the swarm trap. I trapped swarm, that was in the early spring, late winter. That summer, I put a swarm trap out in Clark County, and I was spastic enough, I got a swarm of bees, so I was hooked. 2015, on a Saragram with Penn State, I discovered that my survivor stock, these aren't feral bees now, these are survivor stocks. I had quite a few that I haven't treated for years. They were chewing, mite biting at 30%. And right now, in 2019, they were chewing at 90%. And now, our feral stock in general are chewing about 95% overall, which is fantastic. And last year, there's about 12 of us. We caught 200 feral swarms and swarm traps in known areas of feral swarms. And all these have been tested. They all chew. A lot of work, but we're making progress. And one of the things, mite biting is one of the phenotypes. You're going to see different behaviors that the bees have to keep the mites below 1%. Basically, it's where we're at on the colonies that we're working with at the present time. And our mission statement for the Ohio Queen Bee Improvement Enterprise, it's a mission to exchange information between genetics between the feral trappers, the queen producers, and the beekeepers within Ohio. It's kind of a deviation of the HHBBC and the old Ohio Queen Initiative back in 2012. Megan Melbreath also wrote an article in August 2020 about help, I need a queen. Those articles, both those articles are a must read for all the people that are listening to me tonight. And I have PDFs of both of them, and I'll send them to whoever wants them. Good reads. Tom Seeley mentions in his latest book about the bees, they're finding out that plants and animals, if they're out in the wilderness, and if they run into a problem, they evolve, and within 10 years, they have natural selection that takes care of the problems. And in our case, if you noticed on the timeline between Europe and the US on the mites and how long it took for the bees to start chewing, 10 years. And one of the nice things about all of this, Puerto Rico was overwhelmed with Africanized bees. And a lot of them escaped and they became feral. Within 10 years, the feral bees in Puerto Rico are gentle. That's all research data. Now, we don't want any of those bees brought into the United States, but over in Puerto Rico, they have gentle Africanized bees. So the thing about all this, natural selection favors the drones, and that's what we want. Because if you can get a hive producing 5,000 drones per hive, think about this. If a club has 300 colonies of these bees, and in the perfect world, if each one produces, we'll say 500 or 5,000 drones, look at the amount of drones are in that gene pool flying around, mating with all of the queens in the DCAs, or the drone congregation areas. That's fantastic. Dr. Rob Page, by the way, he came into OSU lab when Rothenbuehler retired. He was the next researcher over there. He wrote a book called The Spirit of the Hive. And basically, he says genetic variation is the grist of the mill of natural selection. He just finished a new book in August of last year. Good book. Some of the books that I read, some of the PhDs get a little bit on the above me technically, but lately I've noticed their writing, their language is getting to the point where a lot of experienced beekeepers will understand what they're talking about. So Rob Page is a good guy. He's out in Arizona right now. Now here's a couple of kids that wrote an article. It's a paper. They used 201 footnotes from around the world. It was just published in June of 2020 about natural selection. And they have this. This diagram is from their article. You see the hygienic behavior, the VH, the grooming, all of that. Their virus resistance, and we have virus resistance with our bees too. By the way, that's been proven by the USDA, and we're going to be doing more of that in the future. You'll see that here in a minute. You see all these different ways that the bees are working on the mites. Yeah, here's the one on the damaged mites. There's a mite fall and chewing behavior. But there's also something on that no one's been able to get the finger on. There's also something happening that the female mites in the cell with the pupa, they are not reproducing. And it's getting better. So all these added up together, these are all called grooming behaviors or phenotypes. The genotypes is the genetics. The phenotypes is what the environment works with the genetics for the behaviors on anything. Human beings, animals, plants, and this is what's going on with our bees right now. This is one of the bee yards up at Honda. I started working with them and one of the engineers in 2019. I set out 3 swarm traps, caught 2 swarms. When Honda came into the US, they purchased 10,000 acres up there in 1977. And their first plant started production in 1982. There's a production plant at East Liberty, and there's a assembly plant on Marysville.
We need a protein feeder for our bees.
In between the two of them, there's an R&D plant. Those are the guys I was working with. We set out 3 swarm traps, and nobody knew we were doing it to see what would happen. Lo and behold, I caught 2 swarms right away. We lost a queen on one of them, but the other one survived. It's number 1 colony, and you can see here, this is their Honda bee yard, and they name all their. You can see there on the right-hand side, they have 10 colonies, and they use the vehicle models for each one of to name each one of those hives. High chewing behavior, and also low mite counts. We have 10 this year. They're going to set out 15 swarm traps in 2021. The assembly plant at Marysville is coming online. They're going to be setting out 10 swarm traps over in their area. And we think there's probably at least 50 feral colonies in the woodlots in that area per the research that Tom Seeley has done. Now, here's the neat thing. I started getting a relationship built between Honda and Dr. Reed Johnson at OSU and Dr. Brock Harper at Purdue in August of 2019 when we had a OPI meeting over in Columbus. Reed talked and gave a talk. I got Reed talked in giving a talk, and Brock came over and was there physically, and he gave a talk. Honda were there. I got them to sit down and say hi and say, maybe we can do something. Well, last Tuesday, I brokered a meeting between Honda and Purdue. Long story short, Purdue is going to be working very closely with us up at Honda on the DNA, and a lot of there's going to be at least 3 research papers come out of this. They're going to be talking about viruses, things like that. And Brock is excited about having access to those bees. And needless to say, Honda is ecstatic. Now, the guys finally got the nerve up to tell their middle management what we were doing. And about 2 months ago, they went to their upper management at Honda of America and explained to them what they were doing. And they were ecstatic. And there's going to be a press release come out after the pandemic is pretty well over. They don't want to compete with the pandemic and everything else that's going on now. It's ready to go. I think everyone thinks about June, they're going to release the press release. And their long-term goal is they want to be able to get different genetic lines up there, and they want to overwinter nukes. And their long-term goal is maybe this year a little bit more in 2022. Any of the OSBA recipients that get the scholarships, they're going to give them 2 nukes of our stock up there, because they want to be able to get their genetics. They realize they want to get the genetics spread out over Ohio. The Japanese are givers. Every year for Christmas, all 3 plants adopt 700 families for Christmas. That's just one of the things they do. They do a lot of work with the inner city kids down in Columbus. Last year, they made, I think, 70 bluebird houses. Honda does a lot of work with kids and the environment. We planted 7 acres of pollinator flowers last year, and it's looking nice. Their long-term goal is they want to have 100 acres of pollinator flowers. With Reed's involvement, Roger knows Harper, McNick Sauter. She's up there working with them on pollen. They're going to have a butterfly way station, and they hope that by the end of this year, there'll be a bee city USA. Is their long-term goal. And the assembly plant at Marysville is following lead. They're going to start putting out swarm traps and working with us. And I got to do some training with them because I did an all-day bee school for them on March the 10th. That's when the door slammed shut for the pandemic. So I treat them, I mentor them. I go up there every week and spend several hours mentoring them on their time. And Honda knows what we're doing. They love it that we're working with the environment. This is really a neat thing. And then we got Brock's kids involved. This is neat stuff. There's Wright Patterson. This is the hop and flying field bee yard on the west end of the flight line. The reason we put bees there was to pollinate the Hoffman Prairie better than it had been. It's 112 acres. It's a remnant of the prairies that we used to have here in this area. And indeed, it worked. The seed production went up high, and then not just on the Hoffman Prairie, there are several hundred acres, about 150 acres besides the end of the flight line is full of pollinator flowers. And talk about seeing butterflies and bumblebees and native bees over there. It was a riot this year, even though they didn't have a lot of rain. They still got a lot of stuff done. Underneath these highs, there are 8 feet of peat. This is a drained fen. Fen is where the aquifer comes up out of the water and cleanses. The swamps or the bogs are the opposite. This is the fen. Cedar Bog, by the way, over north of Springfield is indeed a fen. Water comes off the aquifer up there, and I got thousands of skunk cabbage over there, which bloomed, by the way, in the last week of February. So anyway, so I have 6 colonies here on this end. I have 6 colonies on the other end of the flight line. So I have 12 colonies. They're all feral now. They started out as Purdue, but over years, I didn't replace the queens myself. They replaced their own. I've lost a couple queens, but come to find out, we're having more and more feral stock coming into our swarm traps. And if I have a dead out here, it'll get filled up 1st of May with a swarm, because there's a lot of feral bees outside of the perimeter of the 10,000. There's 9,000 acres at Wright Pat. So isolation, good water supply, good nutrition. Voila, you have feral bees starting to come together for natural selection. So what are we doing? We're going from this ugly picture on your left to this wonderful picture on the right. That's 300 bees that Zell and I did with alcohol and got 1 mite. That colony there is up at the director of ODA's farm and right across the road from Honda. And then here's what they're doing. You can see that the top picture there, one of the legs, is the tip of it has been chewed off. When a mite is injured in any way, it bleeds to death to start with. And then you see the chewed leg there is partially chewed. Then about 18 months ago, I started to notice the bees were getting creative, and they were ripping the intestines out of the stomach through the hole where the rectum is, where the feces come out of the mites. They were going in there and chewing the rectum out and ripping out the membrane that protects the shell of the intestines, and they're eating their own protein, as Barb Letcher says. And then they're starting to mine the tissue around the legs. And this is happening more and more and more and more. It's great stuff. And then you see there the other washes. I think there's 4 mites there on 300 bees that we did over at Zell's place. So that's where the bees are at, the feral bees. And we tested 100 colonies at one time. Long story short, and they were in 3 deeps, either 8 frames or 10 frames, a minimum of 30. They're large clusters, 30,000 bees and above. And when we got done, we only had about 100 mites on the screen bottom board slides. That caught Brock's attention big time. So some of you guys probably have feral bees and don't know it. So here's the 40-hour queen cell history. Larry Connor visited his buddy John Kivas over in France. By the way, John got his bachelor's degree at OSU under Rothenmuller back in the day. Larry wrote about his trip over in France in ABJ 2009. 2010, I was up at his farm and we were having a queen rearing class for 3 days. At the end of the day, we had 40-hour queen cells. So I said, hey, Larry, can I take those down to Data Stallman's place in Blacklick and put them in one of his finishes? He said, sure. I took down 23 40-hour queen cells wrapped in a damp towel, drove for 6 miles, and 17 of the 23 came out and looked very nice, mature queen cells. Joe Lackshaw wrote about 40-hour queen cells in 2011. I have all these PDF files, by the way. And then in 2012, I transported 45 40-hour queen cells in a traveling nuke up to Megan's place at Sand Hill, north of, she's about an hour drive inside Michigan. And Megan has her PhD degree is in epidemiology. She's an expert on Nosema and also on European foul brood. She just got done with the European foul brood last year. I think she had a grant for 3 years from the USDA. So I did that, and then we kind of got busy, and then 2018, I decided it was time to get back into the 40-hour queen cell project again. So 2020, we really started to see good natural selection, and working with Brock and some guys from Penn State, some other folks, it became obvious to us that the feral bees were something that we needed to take a look at. And then in 2020, a September issue was when Megan wrote her article about working with queen cells. That is about a 5-page article, and she goes through from A to Z about queen cells. It's an excellent article. Fantastic. For those of you who haven't worked with queen cells much, we need to get that PDF file in your hands so you can take a look at it. So basic queen biology. Queen lays an egg. This is for a queen or a worker to begin with. It's an egg for 3 days. It hatches into a larvae. It's only, by the way, larvae, eggs, everything else, they only hatch one time. They hatch from an egg to a larvae. They don't hatch out of brood, cell brood or anything else. They emerge. They hatch from an egg to a larvae at day 4. There are larvae from day 4 through about 8, and then the cell's kept, and then the queen emerges in 16 days. In about a few days, about 5 to 7 days, she gets the urge to mate from her hormones. Then a couple of days later, 2 or 3 days, it takes about a day and a half for the sperm to migrate up into her spermatheca. Then the queen starts laying eggs. And then it takes the day 50 from the time that egg was laid until the 49 to 50 days before the brood starts to emerge. It's a long process. So anyway, that's kind of basic queen biology. I just wanted to give you some of you guys, like Emma, some of you guys, you know, this is all new information to you, and I'm overwhelming I'm sure some of this, but I'm trying to keep everybody together. Okay. Upper left, that's about a 24-hour larvae with a Chinese grafting tool. On that queen cell cup, that's not quite 24 hours. And we use, most of us use JZBZ queen cell cups anymore, so we can see the royal jelly eventually. The guy up there in the upper right, I like that age of that larvae. It's probably around 12, 14 hours old. So that's grafting. People hear about grafting. I wanted to make sure that anybody that hasn't been around queen rearing, when we talk about grafting, that's grafting. You graft, and that was a Doolittle. Gilbert Doolittle came up with that idea back in 1880s. He wrote about it in his book. So this is a busy slide, but upper left, those 2 queens haven't mated yet. Dwight's opinion on queens is when a queen emerges in a couple of days, look at her wingtips. If they're at the 3rd segment there from her butt, that's when they're still not mated. And they mate and lay for about 3 weeks. That tail, it's going to be extended another segment. So they're going to have, those are healthy bees or queens, as long as they mate with healthy drones and get healthy sperm. This is one of our cell starters several years ago, this guy here. This is a 24-hour queen cell cup. That's what they should look like at 24 hours, like that. 48 hours, like that. There's no rationalizations. This is the way it has to be. And it works very well. There's the frame that I took up to Megan. Now, we were both laughing when I took that photo. You can look at her eyes, she's chuckling. That worker up there was oblivious as to what was going on. It was still eating some of the protein patty that I had on those bars. And I took it out. They kept on eating the protein. You can see the bees were very interested in feeding those 40-hour queen cells. And in her article, out of the blue, she wrote that article. I had no idea she was. And there was my picture of her doing that. And she explained, I took those up and she got them drawn out. and she gave the queen cells out to a lot of her friends up there in that area. So it works great. There's traveling nukes. That's a CAD drawing of the traveling nuke that I designed in 2013 for our work out at Purdue University. And and those are queen excluders. For those of you who don't know what a queen excluder, those are queen excluders, and I'll talk about them in a minute. When we do traveling nukes, we always have a frame of open-celled honey and some protein and some emerging brood with lots of bees. And what we're finding recently, well, and Jamie and I, Jamie and I were doing our queen rearing classes, and last year when Zell and I were doing queen rearing classes, we had people bring nukes to put the queen cell cups in. In most cases, we found queens in them sometimes, and not enough bees. So Gilbert Doolittle came up with a brilliant idea. You go out and you take 4 or 5 frames of bees, shake all the bees, look for a queen. If you can't find it, shake the bees back into the hive, put a queen excluder on top of the hive, and put the frames in another super on top. Go back about 6 hours later, the bees are going to repopulate all those frames, especially if you have any larvae, anything like that, and no queen. And they're full of bees. So they look like that. Those are good candidates for a small queen cell starter like that. That's what we have, lots of bees. And those are all
Can you repeat that method again?
Pardon?
Can you repeat that method again for how you manipulate those?
Yes.
frames?
Okay. You go out to a hive and you select the frames that you want to make nuke with. In our case, it's queen rearing. So we need to have lots of young bees, and we need to have protein and carbs and young nurse bees. So we select, I have most of my equipment are 8 frame or 10 frame. So when I do this, I select more than 5 frames. I select a lot of frames because I want as many bees as I can get in that traveling nuke. And I shake all the bees off through a funnel, another box, I call it a funnel. Shake all the bees in an empty box, smoke them down, get all the bees down out of that funnel, take the box off, put the queen excluder on, put the box back on, and put those frames back in the box. There have no bees on them, no queen, no workers. And let them come back through the queen excluder with the right age bees on all of those frames. They're a lot smarter than I am selecting bees. They do an excellent job. That way we have no queen, we have lots of bees at the right age. And that's how I make my nukes. I don't look for queens anymore. No way do I look for queens. I do that method, Gilbert Doolittle method, because sure in the heck, I'll miss a queen once in a while. We all miss queens. We know that. Well, if we admit it to ourselves, we miss queens once in a while. This way, no queens in the nukes that we try to produce queens in. Got it?
Yep, makes sense. Thanks.
Okay, sure. If that's a traveling nuke, we use a lot of those out at Purdue University. The reason why we need to have vents on top, vents in the bottom, an easy way to close them up. I just showed the different facets of it. Here's one of the traveling nukes out at Purdue with bees in it. And here's a traveling nuke that's got a lot of inseminated queens. There's probably around $10,000, $12,000 worth of inseminated queens in that nuke. Look at those girls on all of those queens. Always feeding protein patties. Look at the bees on the protein patty. Bees, 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 protein, protein, protein. The right age bees, they take care of the queens. And reiterate one time, Hope is the logo queen for HHBBC. I produced her in 2013 up in Logan County. She was on this bar of queen cells. Decent looking queen cell, full of royal jelly. Now here's the thing, right before they cap at 5.5 days, the nurse bees go ahead and put a lot of extra royal jelly. And it typically extends down into the cup about this far. A lot of people say, well, you know, why you need all that royal jelly? From experience, we know when they cap it, that larvae feeds for 48 hours before it spins its cocoon. So we need to have all this royal jelly, as much as we can pack in there to produce a queen that looks like this. We have a lot of experience doing that. Those queen cells, I wouldn't give you a penny for. Underfed and small. And the reason what happened there, the guy didn't use any protein. And he didn't have enough nurse bees. So that's the thing you got to look out for. Okay, this is a wheel for queen wearing wheel. And I want to take it through here so you understand what's going on. We graft over here at day 4, 24-hour or less larvae in a queen cell cup. And then at day 6, you have a 40-hour queen cell. 48 hours from here to here, we have a 40-hour queen cell. And those are the ones I moved down on a damp towel down to Dana's place for 6 hours. Now, I personally, I wouldn't do that again. I'd use traveling nukes. It's easier. And while I'm traveling, the bees are feeding the larvae anyhow. So I just like it that way. That's a personal opinion on my part. Now we'll talk about these others here in a minute. We're going to move on. There's Megan again, that same picture in ABJ in that article. Now, here's what she did. She took that frame of 48 hours, and I was there when she did. She knew I was coming. She had a finisher ready, and she made up her finisher. She took in her world, it was 10 frames, and on the top super, she wanted to have emerging brood, young larvae, open honey, and protein, pollen. And she had everything running. And beforehand, she knew that the queen was down in the bottom box, a queen excluder, and this super was up above it. She knew for sure that there was no queen in this top box above the queen excluder. And she had pulled out this frame here about an hour before I got there. It was full, and it had a young larvae, a frame of young larvae. She took it out, and she brushed off all of the girls from that frame. And an hour later, that space was just jam-packed with young nurse bees. She slowly put that 40-hour queen cell frame that you saw there that Megan was holding in there. And she wound up with open honey, emerging brood, pollen. Pollen's got to be right beside the queen cell cups. And then you had young larvae. If you have that, you're going to wind up with excellent queen cell cups, just like Joe has there in that bottom picture. Lots of bees, a lot of resources. But you got to do it that way if you're going to have good queens that last at least 2 years. And
Dwight,
lay
I hear a lot of people talking about, wanting to keep all of the larvae out of their cell builder so that the nurse bees have to focus on just those grafts and they're not busy feeding other bees in there as well. Do you have any explanation or kind of insight as to why yours tend to do so well if you have all this extra open larva that may be kind of dividing the labor of your nurse bees?
The young larvae is up there because the young bee, they have young nurse bees feeding that larvae. And we want all the young nurse bees up there we can possibly get up there. And this goes all the way back to some of the Laidlaw. He taught a lot of the guys how to do cell starters. And this is his method, or finishers, I'm sorry. And so does Larry Connor, does this message. And that's the way we do our too. We want to have, I know there's young larvae there, but we have a lot. And also, by the way, we feed them with sugar syrup and we put protein patties. Maybe I shouldn't say this, but I use Ultra B protein, flour and protein patties from Man Lake. It has like 45% crude protein in it. So we flood them with pollen and it works out. And we wind up with good stock, with good mature queens. So now we're going to go to the when the cell is just capped at 5.5 days. That's a few hours before that, they flood that cell with royal jelly before they cap it. Now, what I'm going to do, I'm going to go to the, because I'm going to show you something here. Personally, I love unmated queens in California mini cages. That's my personal opinion. I love it to do it this way. A lot of people in the old days, 15 years ago, said that it was hard to introduce virgin queens to a colony. Wrong. It's not. So you see here rows, you see these 2 rows of cells that have been capped. And before we put them back into the colony for the bees to take care of it, keep them warm. And as soon as they come out, you'll start feeding them. There's a trick with all of this. When you do it this way, you got to put a little bit of queen candy. By the way, I have my queen candy recipe that Joe Lackshaw and Larry Carter gave me. I have it in one of the last slides. You can take a picture of it. It's a good queen recipe. You put a little bit of queen candy here, and put about 3 drops of honey on top of it. So when the queen emerges, first thing she does, she's hungry. She comes down here and gets a sip of honey, and she gets hungry, she works on some of the queen candy. Now, one of the things we've found from experience, we're doing this every 8 hours. We're out here because some of the goofy queens will turn around and want to go back up and start eating the royal jelly. Well, guess what happens? They get their wings caught and they can't back up. So I carry an X-Acto knife with me. And if I find a queen that's got herself in that situation, she's stuck and she's liable to die if you don't get to her. So I cut the queen cell off, and the rest of them, as soon as the queen comes out, we mash the queen cell cups shut so that they can't get up there and get stuck. So you have to do a little bit of extra work, but what happens, you wind up with nice virgin queens. And when you have a good mating nuke set up right, within 30 seconds, you're going to have a stream. I have a video of this. I put that queen cell, or that California mini cage down, and within 5 seconds, I had a steady stream of workers. They smelled the pheromone of that queen, and they had been queenless for 48 hours. They were looking, and they were stuck. They needed a queen. They needed mama. And you can see what they were doing. Now, at this time, you don't know whether they're trying to kill her or whether they're trying to feed her and get acquainted with her. So here's what I do. This is my recipe for successful queen introduction, whether it's a virgin, a naturally mated, or an inseminated queen. I make up my nukes about 48 hours, and then I put queen cage in. But before I do that, I check for queen cells. Now, I'll jump ahead here. And typically the queen cell cups look something like these guys here. And you're going to have maybe 20 of them. You got to kill them all. You got to go and kill all those queen cell cups. And then put the queen in there. Go back 3-4 days later. Check for queen cells again. And you can find queen cells stuck in a lot hidden by the workers, because they get very tricky about all this. But you got to kill all those queen cells. If you do that, go ahead and take the tape off of your queen candy. Now, if you notice here, never put my queens between frames. I have a rim here on top of my mating nukes, and that's where I put my queen cages, so I can see what's going on. That way, I don't have to disturb the bees down below. Then I do this. So 3 to 5 days, I kill the queen's cells, remove the tape. Within 2 days, she should be released, because the queen candy is such, it's not too runny, it's not hard, it's just right. And the queen should be released. I go back in about 5 or 6 days and I check. and the queens will be out. I take the queen cage out. I put the lid back on. I let Mother Nature to take its course. I don't, because you don't know what's going on when that queen's going to start to mate. So you let everything alone for at least 7 days if it's a mated queen, 10 to 12 days if it's a virgin. And this success rate on this is about 95%, whether it's a virgin, mated, or an inseminated queen. It works for me, and it works for a lot of us that do it this way. So that's the virgin, the unmated queens. Now you go on, and on the capped queens, some people like to have mature queen cells, and I've done this. Now, I love working with queen cells, but it takes a little bit extra effort, and this is the reason why. Different ways of transporting mature queen cells. Use a battery box, put some foam in there, and put the cells in there. But they got to be kept at 90 to 94 degrees. And Joe Lakshaw does it this way. I have a Rubbermaid I use, and I put a pound jar of honey jar with hot water, tap water in it, and put the queen cells in, the sealed queen cells. And these queen cells are 2 days from emerging. These are, I'm going to need to talk about that. Back up here. On day 10, you will not touch any of these queen cells. Between day 10, 11, and 12, their legs and wings are forming. And if you disturb them, you're liable to screw up a leg or a queen, or the abdomen gets cooked or something like that. You can lose a lot of queens that way. You don't bother them. But at day 13, I started getting nervous about this time because you don't know, some of the queens may be a little bit early in starting to emerge. So I like to get my queen cells in there a little bit at about day 13 and leave them alone, because, and Megan shows different in her article. She'll, anything that works, to put them in there with some water, hot water. Now I like her idea of taking a hair curler and drilling the cap out and putting the queen cell in there. If the queen comes out and she has, you can see you can put queen candy down there for the queen to, and some honey. So the queen comes out, she can feed herself. Or an egg incubator. I have one of those, works great too. Different ways of moving queen cells, mature queen cells. But then you just, there's the old-fashioned queen castle. You got three frame nukes, work great. I have 2 frame nukes where they work great. Same way. I never put the queen cells between the frames. They're always up here. I can go back there in a couple of days and see after the queen should emerge and make sure they've emerged. Because once in a while, a queen won't emerge. Something happened. I always tear it apart and see why. And I have extra queen cells. I'll put another queen cell in there right away so that the bees don't get too nervous about not having a queen. And here again, I make nuke. I keep telling people how to make a good nuke. This is neat open cell, honey, protein, emerging bees. They're right there. Between those 2 frames, you've got 10,000 workers going to emerge at least in 12 days. But I always look to see, depending on the age of the the bees, if you want them to, a lot of them emerge, you select frames that they're going to emerge in the next day or 2. You can tell by just taking your hive tool and ripping the capping off and see if some of them are going to emerge, when they're going to emerge. Another frame of protein. Something that I don't know, some of you guys may have noticed this or have used them. Acorn out in California are making black plastic triple waxed foundation. Triple is the operative word. We started using these this summer with about a dozen of us, and the results were just outstanding. Now, in my case, my buddy over in Ashland County, where I get my equipment made by Dennis Koblantz. He has two of his buddies. The only thing they make are frames over there. They have commercial equipment, and that's all they do: make frames, make frames, make frames. He can assemble a frame. I think last year they were 95 cents a piece assembled, glued, nailed. And then I take this foundation, the sheet of foundation, triple wax, snap it in, and this costs $1.10 a piece right now. So for a little over $2, it's snap and go. I love it. And the bees like this better than wax foundation, we discovered, even into July. When they start drawing it out, they don't just start up here in the top. They draw, once they get started, we notice they start drawing it all across the frame. I'm assuming the plastic radiates the heat from over here and gets warmer over here, so they start drawing out better because they draw it a lot quicker that we've noticed. So anyway, and here again, I just want to go over this one more time about on the virgin or a naturally mated queen or an inseminated queen. That, put them on a rib, you can see what's going on. And when I don't think I mentioned this, right before you remove the tape off the candy, you pick up that cage, run your index finger across that screen. If all the workers get out of your way, they love the queen. If they're still Velcroed to the queen cage, the screen, let it in for a couple more days. Go back again, run your finger. They're happy with you. If they'll back away, they're not going to sting you. They're not going to hurt you. They could care less. Once you know that the bees love her, you remove the tape, all is well. So basically going through this by kind of a flow mechanism here. Breeder queen, you graft, you transport. Joe Lackshaw, by the way, when he grafts, he's always about 2 hours away from his cell starters, but then he puts them in and it gets to 48 hours. Then you can either it's a little bit more difficult, but I know people that are using 40-hour queen cells and put them directly into nukes. But you absolutely cannot have one egg or larvae in those nukes. It's all got to be sealed brood, lots of bees. You put the 40-hour queen cell in the brood area where they keep it warm. Absolutely no eggs or larvae or it won't work. But I know some guy that he doesn't do finishers anymore. After 48 hours, he puts them in his mating nukes, does it that way. He's done it enough. He knows exactly what not to do and what to do. He's very successful at it. So you can do that also without having to go through the cell starter and cell finisher or whatever. Just FYI. Or you do the finisher. So here's the different types of queens that we see. You never know when a queen comes out in our world what color she's going to be, whether she's a tiger tail like this guy. I love those tiger tails. Now, I didn't take the time to find, I have some jet black queens. These are all from stock derived from ferals. You never know what color the queen is coming out. I have one picture of two queens. One was a red queen and one was a black queen. And they both had the same mother, came out of the black queen. On the other side, we do graft from a red queen and we'll get black queens and red queens. It's diversity. You're going to have that. Nothing's pure. We don't want pure stock. We want naturally selected stock from the feral world. Okay, so now we got these queens all ready to mate. Jay Evans, I love Jay Evans. He writes in Bee Culture magazine, has been for several years, and he calls them essays. He published a book here not too long ago. I would encourage you guys to get that book. It's very inexpensive, and the profits from that book goes into something that Jay doesn't take any money from it. Anyway, he wrote an article in November 2020 issue, talks about the other side, the other 50%, drones, the other 50%. You need to get that. I have the PDF file of that I can send to somebody. So my friends, the Amish, that we get the Acorn Triple Wax Foundation is getting us some drone triple waxed black plastic foundation for us. I'm getting 100 sheets for Zale, and I'm getting 100 sheets for myself. So we can start producing a lot of drones. Somebody had mentioned about flooding the drones in an area. There's, you got around over 2,000 drones, easy, 2,000 or 3,000 drones in that one frame. We need drones, healthy drones, because we have problems with sperm in some of our drones. We know that. We need healthy drones. Sue Kobe gave me these pictures a couple years ago. You can see a drone is mating with that queen. You can see the comet chasing the queen up there. And Mark Winston had some good graphics on a queen, on a drone mating a queen. He breaks off, he snaps off, and he dies right away. That's to prevent inbreeding. So a lot of people have never. Now, one of the things we're doing in some of our projects this coming summer, we're going to be doing some of this work with drones with Purdue. There's some research going to be done. That's exciting about what we're going to do with Honda, with Purdue, because they have a lot of DCAs up there, and we're going to be locating DCAs. But here, if you think about this, if you take a drone, he's got a mother and a grandfather and grandmother. So if you do a DNA on a drone, okay, you have that DNA. So let's say you go out there and you capture 20 drones just by snagging them, they're flying around. And you do the DNA in those 20 drones, and they all have different DNAs. They're from different colonies. Easy way of figuring out what you have around you, just FYI. Feed. I'm doing an article for Bee Culture Magazine about putting feeders out. I'm almost done, Roger. One of the things that I noticed through the years, around the 1st of January, we put out protein feeders, the commercial guys. And then I saw this over in Pittsburgh many years ago. Somebody had one made up. These are 4-inch thin wall PVC feeders. And I don't have a complete picture of one. The article probably will come out in about 3 months. I have pictures. I guess I should have put a picture. I have a picture of Wyatt putting one up for me over at his place about a month ago. And we feed, this is out at Wright-Patterson, I believe. No, this is up at Honda. But what we're finding is after the last hard freeze, absolutely everything's killed. We put these out and the bees were all over them. They were telling us something. Even though we had a fairly decent fall as far as pollinator flowers, both protein and carbs, they still needed more flower. And every day at about 43 degrees, sunny day, they're all over these feeders. Then we started feeding some protein patties about a month ago, and to some of the late swarms. And they're eating the protein patties, not like they have in the past. There's something going on with protein, at least down in this area, just FYI. Now, here's the other thing. Back in the day, Tom Seeley did a lot of tree lining to find out where his bee trees were up in the Arnott Forest. And he wrote a book about it. And I have a tree liner, but I'm lazy. So what we're doing now We learned this trick a couple of years ago. If we go into an area, we don't know what we don't know about, and it's isolated. We don't know where there's feral bees there. Well, we put out these feeders in an area where it's sunny at about 45 degrees. If we see bees there, we take a picture of it. We look at the diversity and the color of the workers. And we see that from experience, we know to put a swarm trap in that area. Been extremely successful in doing that. Now, here's the thing with swarm traps. I've got a presentation on swarm traps by itself. When a scout bee is out flying around trying to find a new home, while they were still foragers, when they're out foraging, if they see a hole in a cavity, they remember that. And if they're selected to be one of the 500 scouts to go out to find a new home, they'll come back and check this out. I've got videos and I've got photos of, because of the work I did up at Honda, of the scouts flying into our adapters on the bottom of our swarm traps and going through and measuring on the first day that they find it. And then shortly they'll have another 2 or 3 bees that they go back and tell them, come over and, hey, check this out. By the end of if they're successful, by the end of the 6th day, they'll have several hundred bees there and checking things out. They fly back and the swarm's about ready. Those bees are the ones that brings the swarm over to that location. And all that research has been done by Seeley and somebody else. So double screens, one last thing, I think. Double screens. You know, a lot of people have got to buy packaged bees. They can't help it. There's not enough nukes in Ohio. And if people want to get into bees, you got to buy packaged bees. I understand that. So this is a snell grove screen that my son makes me. He does woodworking for a hobby. This is one I've made just with a hole saw and put screen on. Both are very effective because what we're going to do, let's say we have a substandard queen and a very strong colony. This colony is full of bees. There's a queen in there someplace. First thing you do is put the new queen with a nuke above the double screen on the hive. Let it grow for a month, 2 or 3 weeks, whatever, until it gets fairly strong. This is a SARE grant that my buddy Jim Kearns up in Logan County did several years ago on a two queen system. And this works great, whether it's a 5-frame, 8-frame, or 10-frame. And you allow the nuke to grow until it's strong. You go out, set the top and nuke aside with the screen, and put the screen aside, the double screen aside. And if you have 2 boxes there or 3 boxes, whatever, you put a queen excluder between all the boxes. You go back in 4 days, you find eggs in one of the boxes. That's where the queen is. You don't need to find the queen. You just know that the queen's in that box. So you set her aside. You put a newspaper on the brood nest. The other 2 supers, the 1 super, what do you have? You spray some sugar syrup on and put your nuke on it. Put the double screen above the new queen and put the old queen on top of the double nuke. Let her lay for a while. You have 2 queens laying in parallel. And if it happens to be a weak colony, in about a month's time, you're going to have a strong colony. Or you take that nuke, you take it away and make nukes from it. Whether you kill the queen or whatever you don't want to do with the queen, I don't care. You can make nukes from that top nuke with the old queen. Make more nukes. You can make nukes from nukes from nukes. You go crazy making nukes with lots of brood. And there's an example of an 8-frame above the double screen. That year I had 9 going into winter. All 9 of them survived the winter. Great way of overwintering nukes, new queens for the next year. Another picture of it. I hadn't pulled the tape off of the for the vent hole for the lower colony. And then I get, this is my last slide. I promise you faithfully, Roger. Mike Palmer, good friend of mine. double nuke systems. Those are 10 frame bodies with 3/4 inch wood in the middle. You have 4 frames this side, 4 frames this side. Queens cannot meet each other. In this case, we put cantilevered, and so we have 5 frame nukes because I have 5 frame supers. So you have 4 queens there. Now look at this. On April 1st, we start out with a 5 frame nuke. By May 21st, you had brood here, 4 frames of brood here. You always have a honey frame here. 4 frames of brood here, 4 frames of brood here, a couple frames of brood here, honey, honey. Look at all those nukes you can make. And we have done it that way. Or you overwinter nukes that way. Because if overwintered nukes this way, they'll form a cluster between the two colonies. And there'll be one big cluster like this, going over winter. Next spring, you better hurry and get out there and put supers on it because they'll swarm on you real early if you don't. You got to work those every 7-8 days. But look at all the brood you have. If you're looking for brood, one of the things Mike used to complain about on goldenrod flow, he would have swarming, and he had too much brood. He was overwhelmed with brood. He would take a super, a 10 frame super, and fill it full of 10 frames of completely capped brood to put on weak colonies, then put 2 supers above it for a honey flow. I mean, think about that. 10 frames of brood emerging in 12 days. My goodness, you'll have bees everywhere. So anyway.
Dwight, when you say you have to work those about once a week, what kind of stuff are you doing? Are you just adding more 5 frame boxes to them? You're harvesting brood? Or?
Yeah, harvesting brood, making sure they have room because all the heat, it's a heat thing. Heat this way, and there's there's a little bit tight. Oh, here, we'll pick on this one here. It's tight. So you have heat transfer this way, and there's always brood on the inside frame of this frame next to this super. Always. She's laying her heart out. There's heat. And you put a new super on top. I've had them above my head. It goes crazy. Then we start using brood, making more nukes. So I call them brood mares. And that's what Mike overwinters 750 of these systems every winter. And he sells a lot of nukes. So there's a queen candy recipe. And there's only one thing I got to let you, and I mentioned it here. I use a cup of honey for our own use, but if you're going to be selling this queen candy in queen cages or whatever, by law, you got to use either Numaline or use corn syrup, not the honey. This recipe is perfect. And I use a microwave, and I have a temperature sensor. And when it's pliable at 95 degrees, and I can pull it up like taffy, it works great. Because you got to be careful. If you don't watch yourself, you'll have sticky queen candy, and the queens will get mixed in with their wings and they'll die. If it gets too hard, it works the other way. But this works great. I have used this recipe and used the microwave on it many times. And Laidlaw was the one that taught Larry Connor this, and also Laksha. So this is a proven recipe for queen candy. Recommended reading. For you experienced beekeepers, this book here is a great book. It came out about 18 months ago. Here's Jay Evans's book on his essay. He calls them his essays. There's Dewey Caron's book on honeybee biology. Rob Page just came out with this book a few months ago, The Art of the Bee. That's an excellent book also. And this book here is the average beekeeper will read it, and there's no very little scientific language in it. It's all very well easy to read. And of course, the bee magazines, both Bee Culture and American Bee Journal. For those who haven't made nukes before and want to understand queen rearing and all of that, this is a good book just on bee sex and the mating biology of honeybees that Jamie Ellis and Larry did for the Chronicers. That's an excellent book on mating biology. So that said, this is the last slide. We're going through a paradigm shift. That's one of the things that Zell and I keep talking about. Change is difficult for a lot of people. We understand that. But I'm hoping that this presentation tonight, you get a feeling that this isn't just some dream that we're dreaming up. All this work has been done many, many times. There's nothing new here. We have been working with this for some time. And now that we have Purdue working with us, Purdue's going to spend a lot of time with us this year in Ohio with me and then also with Honda. This is fantastic because we don't have, Reed Johnson, bless his heart, he's a scientist. He's not a beekeeper. And we don't have a Rothenbuehler like we did in the past. And we don't have a extension of beekeeping. So, but we have someone that is willing to come over with his people at Purdue and work with us. That's neat. So that's it. I'm a little bit late. Sorry about that, Roger.
No problem, Dwight. If anybody has any questions, go ahead and unmute yourselves. Ask Dwight any questions. I'll start things off. You mentioned something about your pollen patties, that they're not eating them as much. Is that what you were saying?
I'm saying this year they're eating them more than they have in the past.
Okay.
Now, typically, I haven't putting patties on until about the 1st of February. I don't want to get them built up too much and swarm on me. But we noticed for some reason on a hunch, because of the way they were treating the dry protein feeders, we thought we'd try it and see. They gobbled them up very quick. It wasn't just a gradual, slow feed. They were gone in a matter of a few days. They jumped on them right away. Now, the queens are laying eggs. They started laying eggs about December the 27th, 28th down here in this area of the country of Ohio. So you have full-blown egg laying going on right now. So they need protein to feed that larvae. So it's just something that we saw this year that was a little bit different than past years.
Do you feed pollen in the fall?
I feed personally on my swarms and my nukes. August the 15th, I start feeding protein patties during the time that they're making winter cluster bees. Yes, even though protein pollen's coming in, they gobble those. I put a half a patty on to start with on a 5-frame nuke because a lot of my swarms go into winter in 5-frame equipment. And if they gobble them up in 7 days, I start gradually opening, putting more and more on. And during that time, they're making as many winter fat bees, winter cluster bees as they possibly can, because that's what's going to live from about 5 months, 4 to 5 months, to get them to the point where they're going to start dying here next month. So that's why the queen's starting to lay now to replace the winter cluster bees that they made in August. So yes, I feed. The stronger colonies typically have enough protein from the pollen, depending on where you're at. Where I have my bees, they have good, I try to keep my bees in good natural nutritional areas.
I tried one thing, November, early December, we had like a week of 75, 80 degrees, and I put the dry pollen feed out in 2 yards and nobody touched it. So they weren't raising any young then, and they didn't want to eat it. So they know some of my yards, they're not touching the flower. Most of them are, but I have a couple of yards that don't, but they're in good above average nutrition. So the assumption is they have plenty of pollen. Correct. Good.
Right. That separate feeder behind the Honda hives along the tree line, being low toward the ground, is that an issue with varmints?
No, Oh, I didn't know. I don't know whether you saw it. Yeah, I think you did. I use 1/2 inch screen on all of my feeders to keep the birds out and the squirrels out. But no, to answer your question, no, they don't bother them.
I think usually the bees are so busy in there, nobody else is going to bother them.
And we have plenty. The other thing is up at Honda, they have a lot of deer, like right, Pat. I mean, lots of deer. And we see deer all the time up in that yard right there in the woods. And they've never bothered anything. We do have all of our colonies staked because we had a lot of young bucks up there this year. And we wanted to make sure that they didn't rub up against our beehives, because we have had that happen many years ago. I learned a hard lesson real quick. So I stake everything that's above 2 deep, 5/5 nukes. Most of my swarms go into winter 3 high or 4 high. And they'll come out of winter just raring to go.
Hey, Roger,
Yes,
And-
sir.
and when you drive into Honda, there's a sign saying there, the animals got the right of way. Just think if the state of Ohio would do that, there would be about 35,000 deer wouldn't get run over.
Yeah, there you go. There you go.
Yep. Honda is very protective of the only thing they don't like is the Canadian geese.